let's talk about an architecture of lymph nodes where the antibodies are built to be very specific. So when what happens is all of our tissues there are kind of like washed with liquid, with water that seeps through our blood vessels, and along with this water, interstitial fluid is what it's called, also come immune cells, and this way they can wash the entire tissue and scan tissues for any invaders. So for example, if you get a vaccine in a muscle, or if you get infected, within minutes to hours, the patrolling immune cells can recognize this antigen, and then they bring that information to lymph nodes. So eventually all that liquid starts collecting, starts pooling, gravity takes over, and they enter lymphatic system. So that interstitial fluid eventually becomes lymph fluid. So it gets concentrated, and then your lymphatic system, which has its own network throughout the body, travels through, and eventually it reaches lymph nodes. And lymph nodes is where you organize how antibodies are made. So let's talk about the composition of lymph nodes. So there's primary follicles, and primary follicles contain naive B cells. B cells are the type of cells that eventually will be transformed into plasma cells, and plasma cells produce antibodies, okay? So primary follicles is like a warehouse for B cells that are virgin. They haven't been transformed to anything yet. They can be used for any purpose. And then next to that, we have secondary follicles. Now, secondary follicles form what is referred to as germinal centers, and that's where you start honing B cells into producing specific antibodies. So these B cells will be transformed into what is referred to plasma cells, and it's actually the plasma cells that eventually then form specific antibodies. So this is where you start making antibodies target something very specific. This is also, germinal centers is also where you can start switching antibodies from one form to another. For example, from IgG1, which is one of the early antibodies that can be specific, to subsequent forms like IgG3, IgG2, and then the last one is IgG4, and we'll obviously talk about IgG4. So that's germinal centers and secondary follicles. And then we also have extrafollicular zones. And extrafollicular zones is where B cells can be transformed into plasma cells, but outside of germinal centers. And the reason why that happens is so that they can be transformed into plasma cells that will produce antibodies very rapidly. And the reason why you want this is so that you have a very rapid response. So what happens in a lymph node, in the secondary follicles and germinal centers, it takes time. And then extrafollicular zones outside that secondary follicles, that takes place really, really quick, so that if we get infected or get a vaccine, the response can be much more rapid before antibodies get to be honed in to be more specific. All right, so think of it as like it's like a place where B cells can have different career paths, right? So they can go either germinal centers or those extrafollicular zones. Now, let's talk about two types of antibodies that can be formed, two types of cells that can be formed in that entire region I just told you, okay? So first, let's talk about short-lived plasma cells. So those are formed in those extrafollicular zones, and these type of plasma cells will produce short-lived antibodies. These antibodies are not very specific, not super precise, but they're very rapidly made because they made in those extrafollicular zones. So the idea here is that you want to have antibodies that are instantly available, but they are not super specific, so they just want to start managing the infection without yet making a precise response, but at least start the first management. So those are short-lived plasma cells, okay? And then the other option is long-lived plasma cells. So remember, plasma cells are B cells that become plasma cells. Plasma cells are at the end responsible for producing antibodies. And then long-lived plasma cells, those are made inside those germinal centers. So when a B cell enters germinal center, it has two options to choose from. It can either become long-lived plasma cell, which will then long-lived plasma cells then go to bone marrow and they reside there. They can even reside there for almost your entire life. And they will for the rest, potentially for years or even decades, depending on the type of cell, they can produce antibodies for even sometimes duration of somebody's entire life, or years. Sometimes it's shorter. It depends on the type of antibody. So that's why they're called long-lived plasma cells. But a B cell can become that, or it can just become a memory cell. And a memory cell, it means, oh, okay, we had to respond to this specific antigen. So we had to respond to this specific area of the spike protein. Let's put that in a memory. And then memory B cells, they don't go into bone marrow to produce antibodies. They are put back into tissues and start monitoring so that if the antigen, let's say another spike protein comes in because of second, say, vaccine or third vaccine or an infection again, they are now primed to respond very specifically. So those are the two types of plasma cells that you produce. You produce memory B cells, or memory B cell can also become short-lived plasma cell, which will start producing antibodies really rapidly, but low affinity, not very specific, or long-lived plasma cell, which will produce very specific antibodies and will do this job for a very long time. So here's the interesting part. The antibodies that either short-lived plasma cells or long-lived plasma cells produce are the same antibody, but the sugar coating is completely different. And it's the sugar coating that completely determines its fate, what it will do, and its function. So let's talk about that. Let's talk about the sugar coating now. Antibodies are actually glycoproteins. So these are glycoproteins are proteins that are coated with sugar. Spike protein, by the way, is a glycoprotein. It has sugar modification or decoration all over, and so do antibodies. And there's variety of different sugar molecules that can be attached to the antibodies, and depending on the pattern of the sugar molecules, it will determine. Not only does it tell you where the antibody came from, because it will tell you that the antibody came from either short plasma cell or long-lived plasma cell, but it also will determine how it behaves afterwards, meaning how the immune system will respond to that antibody. So let's talk about short-lived plasma cells, because those are the earliest ones. So short-lived plasma cells can start producing within first few days. The most famous short-lived plasma cells are, I think, IgM antibodies, and sometimes it can already be IgG1 antibodies, right? But like I said, they're not very specific, and these short-lived plasma cells, they have very high galactose. Those are names of type of sugar molecules. They have high sialic acid, again, another name for a different type of sugar molecule. They are highly fucosylated, so fucose is another sugar molecule, and they are not branched like they could be as well, but they're not. So, and branching means branching of sugar molecule patterns, all right? And that pattern overall ensures that immune cells with the receptors will not recognize these type of antibodies well. And as a consequence, these antibodies, when they attack the pathogen, let's say virus, and they recognize the spike protein, it means that the response to these antibodies, body immune response is very gentle because the immune cells will not be recognizing it and therefore they're not responding. So they're not cleaning the pathogen that way. What happens is what will respond is something what is referred to as complement system. And complement system, think of it as like a series of proteins that are naturally floating in our circulation, and they're always there. So it's part of innate immune system, and they can immediately respond and signal to different arms of immune system, or on its own it can start destroying the pathogen as well that is attached to the antibodies. And the reason why we want this is because that initial reaction by short plasma cells is just to start controlling the problem, which is the infection, without really introducing high-level charge cavalry that was potentially could start destroying. We want to make sure we give some time for antibodies to mature, to become more specific. And only once the antibodies become specific do we want to make sure that then we use extra forces to clean out the problem. Because if we didn't, we would run the risk that we could cause too much injury in our body. So that's why the short-lived plasma cells with their antibodies, short-lived antibodies, that's why it's more of a gentle initial response. And then within a week to multiple weeks, when germinal centers are finally formed and antibodies can start being more zeroed in to be more specific and target more specifically, that's when those antibodies have completely different sugar molecules. So now they have lower amount of those galactose, they have lower amount of those sialic acid sugars, they are afucosylated, meaning they have fewer fucose, and they are more bifurcated, or they have more of these branches introduced into them, and that completely changes how these antibodies are then recognized. So same antibody, different sugar coating, completely different outcomes, completely different behavior of these antibodies. And then what happens? So what happens with these long-lived plasma cells that produce these long-lived antibodies with these altered sugar molecules? Now immune cells, such as natural killer cells and neutrophils, they now recognize this, and they can really respond and really attack the problem. And it's okay now because the antibodies are more specific. So now the attack is more zeroed in, and it's less likely that we would cause tissue damage or problems. So natural killer cells, they will target very specifically, and neutrophils are very interesting. Neutrophils, they are like kamikaze cells, and they can literally burst open and release their DNA, and the DNA acts like a mesh that can trap pathogens. So they're very unusual, but both of these events are highly inflammatory, and of course, if you have a lot of inflammation, you could run into a problem, which is why we want specific antibodies to induce this type of inflammation. So you can see there is a difference how antibodies, depending what's happening with the sugar molecules, will have completely different behavior on the immune system response. And also the sugar molecules is like a name tag telling you where they came from. So that's the background.